Thank you so much for the invitation, and also thank you so much for the introduction. And it's a great honor to be here. Actually, this is the first time I came to Suzhou. I love this place. Thank you so much. And actually, I have changed my title and also the content a little bit. I think the title, actually, I have used the old title for like 3 years. So decided to revamp the slide deck for this. And so now I will talk about the past, present, and future of the pangenome, mostly the human pangenome. First, where to start? The pangenome, this terminology started, I think, around 2003. And at that time, the first publication mentioned pangenome, this word, is in this one. And that time, that was like more than 20 years ago, the pangenome was first used for the total sum of genes of the living organism and also the viruses. And this is actually quite different from the pangenome we are using. In that paper, the pangenome means the whole, the everything, every living organism. But currently, the modern definition of the pangenome happened actually is 2 years later. That's in 2025. And this is the PubMed publication. If you if you use PubMed and you search for the pangenome these words, and you will see recently, especially in the last 3 years, you can see a spike of the pangenome work in recent literature. And this roughly shows the timeline of the pangenome work. This, as I said, is a biased timeline. This is now really a complete one. And so in 2005, 2 papers, but both of them coming from the same group, defined the pangenome. And this is actually the pangenome fairly close to the pangenome we use today. And this is the pangenome still used used for the bacteria species. The definition is like this. The pangenome is in that paper, in the 2 papers, the pangenome is split into 2 parts. The first part is called core genomes. This means that the genes present in essentially all the strains. And the second part is called the accessory genome. These are genes present in 2 or more strains, unique to or unique to single strains. And here, it is important to know that to annotate the genes, you really need the genome. You cannot use a bunch of short reads. If you just use short read, you cannot annotate genes. And so, if you have the reads, you have to assemble into genome to make the to make the genome annotation. And it's important to know that the bacterial pangenome is really a pan-proteomes, because if we use this particular, we are focusing on mostly the protein coding genes for the pangenome. And the basic procedure to construct the bacterial pangenome is like this. Basically, we have a protein list of the bacterial genomes, and then we do gene annotations, which is usually easy because the bacterial genome don't have the introns, and so you just look for the operating frames, and then you can annotate the genes. When you have the genes, you do clustering. You can cluster all the genes, and then inspect the cluster to find the orthologous groups, and this will be the pan. And for each orthologous groups, and so you just check whether this group of genes present in all the species or in part of the species, and you can define core or pangenomes. There have been tools for this. This Rowry, this is one of the earliest, and these days still actively used, although it's not that good these days in today's standards. And the pangenomefrom, they published 4 years ago. This is a fairly recent one. And I think after that, after pangenomefrom, there are very few papers on the pangenome and on bacterial pangenome. And how well these two work, and this is on these hundreds, MPB genomes. And you can see for these 3 different tools, the pangene I will talk about later, pangenomefrom, the current one of the best tools, and also the pangolin. And you can see most of these tools, the 3 tools are quite agree with each other very well. Out of the 3,400 genes, there's a large overlap. And this is the core genome. As I said, the other part is the pangenome, the entire genome. And you can see these still agree well with each other. The pangolin is a bit different. This is mostly because caused by the frame shift. In the tuberculosis genome, and there are some frame shapes, and this is creating new genes. And so this depends on the definition, what is the protein coding genes. If they overlap on the genome, are they the same genes? And if they are caused by the frame shape, are they the same genes? There's no universal agreement on that, though. And so this pangolin prefer, if there's frame shapes, you still count the new genes. But for the other two tools, if they have a large overlap on the genome, they will think, these two tools, you will think this is the same genome. This is the difference caused by, but still, these agree well with each other, these three tools. And this is a more complex case. This is, although there's 50 the E. coli genomes, you can see the agreements very well. They agree with each other. And the pangenome is also good. And you can see this pangenome is much larger than this one. Just go back to this slide. You can see the pangenome and the core genome are about the same, which means this MTB has a very tight pangenome. I mean, in the bacterial world, it's called a closed pangenome. And this is people usually call it an open pangenome because this number is much larger. Bacterial pangenome, as I said, the pangenome is mostly a pan-proteome. It's not really the whole genome. It's about the protein coding genes. But for bacteria, it works well because bacteria, more than 90% of the genomes are actually protein coding genes. And so this works very well. But this doesn't work really for animals or plants. For example, in a human, there's only 1% of the genes are protein coding genes, I mean the protein coding regions. And the procedure I talked about doesn't work for the animals or plants, mostly because the gene annotation becomes much harder with splicing. In a human, there's alternative splicing and all sorts of things, and it makes gene annotation much harder. And also the human genome is 1,000 times larger than the bacterial genome, and there's also the scaling issue. And so recently, mostly actually this year, I mean, my group developed this pan gene. Is basically migrating these bacterial ideas to the human genome. And the procedure is slightly different. It aligns the protein sequences to each genome to do the gene annotation and identify the orthologs based on the overlap between the genomes. And I will repeat it using this gene as an example. And this C4 gene, this is one of the strongest keys to the schizophrenia. And this gene is a HRA class 3 gene. This gene has 2 copies. In the current human reference genome, there are 2 copies. There's the C4A, this is copy 1. Actually, both genes have this gene structure. This is a very long gene, 41 exons. And between the exon 9 and 10, there's a transposon insertion. Some gene has this transposon insertion, some gene don't. And the 2 genes, C4A and C4B, differs on the exon 26. There's 4 amino acid changes, the 4 key amino acid changes on the functional domain that separates the two genes. And in the human reference genome, this is the AL means C4A. Long form means there's this HRV insertion. The short form doesn't have the HRV insertion. And the reference genome has this. The ALBL, this means this C4A long form, C4B long form, both have this HRV insertion. And you can see in the population, there's the short form. There's some people only have 1 gene, and some people have 2, 4, 3 genes, and in the rare case, there are 4 genes in here. And so this is what the gene looks like in the population. And if you run the pan gene on the human assemblies, on the 100 human assemblies, you can see this roughly capture this. You can see the most abundant haplotype is C4A, C4B. Here you can't see the long or short form, but at least you see there are 2 genes, C4A and C4B, the most abundant, and so this is the same. And so there's most people have two copies, and some people have 1 copy, some people have 3 copies, and there's also other genes around here. And this captures basically the different haplotypes between the human within the human population. And Chaolin recently have a paper, I think, in Cell, as you remember, talking about this MAPT gene. This is has an association with Alzheimer and Parkinson. And you can see in this gene, the MAPT, most of the people have this gene on the reverse strand, but some people is flipped on the forward strand. And also you can see other, for example, this gene has 2 copies in this haplotype, but 1 copy in this haplotype. And you can see there's a variety of haplotype diversity within the human population. And of course, the HR genes also have this. The HRA genes has, for example, some has this DRB3, some has 4, some has nothing, and some has 5. But we can do, on the HRA gene, we can do a step further. Because for the HRA genes, we also have in addition to the gene itself, the absent presence of the gene, we also have the allele groups. And so here, usually the naming is like this is the DRB, HRA DRB gene, and star means the set separator, and then the 0, 1 allele group, 1, 0 allele group, this 3, 11, 12, these are the allele group. And if you annotate the genes on the HRA haplotypes, you can see the correlation. There's a very strong link between the allele group, the DRB allele group, and the presence or absence of this 3, 4, 5, or I mean, I have no gene here. And this is, you can annotate the gene and study the haplotype structure. And there's a more complex case. This is the HRA, around the HRA genes. HRA gene, this A gene, you can see the allele group associated. Some people have this, although these pseudogenes, but this is a long structural variation, it's a very long deletion. And some people have these 4 pseudogenes, some people don't, and similarly this Y gene. And these are all strongly correlated with the allele group, but not perfectly linked with the allele group. This DRB case is perfectly linked to the allele group. And if you look at this, I talked about the C4 gene, this is class 3 gene. C4A is a class 1 gene. This DRB, this class 2, HR class 2 genes. And you can see the gray means that this gene is absent. Okay, each line, each row is a haplotype. And so if you see a gray, this means it's absent. The gene is absent on the haplotype. And you can see these 4 pseudogenes. Some people don't have this, and it's HRY. And similarly, this DRB345, there's the changes and the C4 genes. And also the heat indicating the average diversity, average divergence from this haplotype to the average haplotype. You can see there's for these genes, the divergence is like 5%. And in average for the other human genes, that's about 0.1% that divergence. And this gene, the divergence is 50 times higher than a normal HRA, I mean, this human gene. And the HR genes, you may say the HR gene is complex, but it's actually not the most complex, and even this is not the most complex. And there's here this killer Ig-like receptor regions. These are a list of genes, about 100 kb, having this list of genes, and they have a higher diversity. The most abundant haplotype is this one. Most people, this is called A haplotype. Most people have these genes, but you can see some other people have actual genes. Some people have duplications. For example, this part is duplicated, this part is duplicated, and for these are different. And the killer gene, these haplotypes are more complex. There's also other HRA killer. So this is also an immune-related region. This is TCR, BCR, the T cell receptors, B cell receptors, and basically the antigen region. And previously, people do a lot of studies about these regions. Most people use the RNA-seq to analyze the expressed form. But now with the assembly, we can study basically the genomic region. Previously, we can't really do that because these regions are much more challenging to assemble than the KIR or the HR regions. And so now with the assembly, we can study these. You can see these regions have genomic variability as well. For example, this IgH, this Ig heavy chain region, you can see here the constant genes. They are actually this not just linear. They have multiple genes. Some people have some C genes, some people don't have some C gene. And you can see this also they have the structure of this. This is annotated with a new tool. This analog hasn't been published yet. And if we zoom in to this small section, you can see the haplotype. This is a different haplotype. This is a count. And this is a different IgHV segment. You can see this. There's a gene duplications. For example, here you have this gene present 3 times. The V342. This 42 duplicates, you can see this type has 3. But for most of these others, for the V3 42, there's only one copy. There's a but on the genome, these segments are already varying. And how that's correlated with the expression, and so far actually we just have this tool working, and so we don't really know. And just I show this example that the human genomic region are actually highly variable, especially on these immune-related regions. Okay, I have shown these kind of the interesting genes, but there's the most of the human genes are actually kind of non-interesting. And so here is the number. There are 14,000 multi-exon genes. I mean, this is counting the pseudo genes because sometimes it's harder, but there's the single exon genes, some of pseudo genes, and so sometimes it's difficult to tell. And so I'm only counting the multi-exon genes. And for among these genes, only actually less than 0.5% are missing, but has a presence and or absence variations in the human population. And only 13 of them, 13 of the 53 don't have paradox, of which, 4 are these KIR or HR genes. These are real cases, and we know that has happening. And the other cases are all in very complex region, or this. But anyway, of these 13 genes out of 16,000 genes, that's less than 0.1% of the genes are actually having the real copy number change. Basically, they can absent or present. And for the vast majority of genes, if they have copy number changes, these actually they are still present in the human population. It's just that you have one more copy or one less copy. But the gene content reflect the changes. And so these come to the research question that is the pan-proteome, this approach, this mostly learned from the bacteria, that it really works well for the human, in the human case. It's still useful, but it's limited. There's several problems with these gene-centric approaches. The first is that there are structural changes to the genes. Just before talk, someone asking me, have we thought about the pan-proteomes? And we are thinking about that. And for these genes, we may have actual exons in the genes. There's one example, this LPA, there's also CR1, there are several cases like this. Basically, in the human reference genome, you have one gene annotation, but in a different individual, it may have actual exons in the genes. And so you cannot really annotate them. And with this gene-centric approach, you cannot detect that because we assume the gene, everyone has the same gene structure. And secondly, only 1% of human genome is coding rich regions. And so this, for bacteria, that's like more like 90% of the genomes are coding rich regions. And so if you only focus on this small fraction of the genes, we don't understand, say, the transposons and also the other more complex rich regions. And the third reason is that the gene content of the human genome is very stable. And so then that's the risk question, whether it's worth studying this, but this only contribute a very small, there is an absence and the presence of novel material only contribute to a very small fraction. And so we need to understand the rest of the genome. And so that comes, I call it a real pangenome, because I said the pangenome was, meant to be for the bacteria. There's really pan-proteomes, and so the pangenome represents the genome sequences. And in the modern era, and what's the pangenome mean? I usually take this definition. A pangenome refers to a collection of assembled genomes in a clade, typically a species, to be analyzed together. And bacterial pangenome still counts because it's an assembled genome and usually applied to a clade. But the realignment and for the Western genome don't count as pangenome because it doesn't really have a genome. It's just a bunch of short read and variant coding. Although a few review papers still say Western genome is pangenome, but I wouldn't count that way. And so this comes to the human pangenome projects, where I'm also on the steering committee of this. And human pangenome projects, we also call it the Human Pangenome Reference Consortium. This HPRC, I will talk about the HPRC occurred later. So this project started in late 2019, and it is the first bacterial result published in last year. And in that paper, we assembled 43 diploid genomes. This data are fully public. You can use for any uses. And this is driving the development of different algorithms. And so one question is why we start to analyze, to do this human pangenome in 2020. We have bacterial pangenome like, 20 years ago, but why we started this now? A key reason is that we can really do the assembly. For bacteria, because the bacterial genome is small, and so we can basically use Sanger arrays and even the shell arrays, we can do fairly good assembly of the bacterial genome, but not for human pangenome. And for human pangenome, and you can see this line, there's a different, on the x-axis, it is different assemblers. On the y-axis, you just think about this, the error rate. This is the number of multi-copying genes are missing. Most of the previous examples, C4 and this HRA genes, these are fall in this category, but they have multiple copies. And you can see for the older assemblers, sometimes the 90% of the multi-copy genes are lost in assembly. And if you lose these genes, then you are losing the most interesting part of the genome. And so can't really study the pangenome. And so, but with the more recent algorithms, and you can see the error rate is only 0.3%. We can assemble almost all the multi-copy genes. And so this gives us the chance to study this gene. Previously, it's not. And this is mostly driven by the accurate long rates, the high rates. And very briefly about the modern assembly process, it generally looks like this. You start with the race. There can still be sequencing errors on the race, and so do the error correction. You correct away the remaining sequencing errors, and you do the initial assembly graph. And there may be tangled some regions you cannot really resolve. There's some regions you have gaps, and so use another type of data that's called ultra-long race, and you can do the assembly, and then use the long range data to fill in the gap. And this way you can do the assembly. And for the diploid genome, the procedure is the same, although the graph will be very different. And still you start with the race, and so this time you have the heterozygote size and between the two haplotypes. And still you correct away sequencing errors and you do the assembly graph, and this assembly graph usually has this, we call the bubbles, have this special topology. And then you use the ultrasound data to resolve these bubbles, and then finally you can get the assembly. And with the modern assembly, we require the accurate long rates. The mainstream data type for this is the Bacterial hi-fi race, and there's also very recently, just earlier this year, people has make the nanopore working, but still this is a working in progress. And typically these days, if you do this high quality assembly, it costs a few $1,000s. It depends on how many data you want and how high quality you really want to achieve. This can be assembled in one day, and each of them these days, each of them is much better than the current human reference genome. And you can do that in one day with a few $1,000s. And this type of data make pangenome possible. As I said previously, you can't really do that because you cannot, if you cannot do the assembly, you cannot recover the multi-copy genes. You cannot really study the pangenome. And after we have the pangenome, what do we do with the multiple assemblies? I think the popular approach these days is called we use the pangenome graph, we use the box graph approach. Basic idea is in this example, we have two genomes, and we want to construct the pangenome of the two genomes. And so the basic idea is to collapse the identical region. For this region identical, we collapse them into these nodes. And so this will become an indel between the two genomes. And so this part is identical, collapse them. You just collapse all the identical sequences. And in this graph, a node is a sequence, and the edge indicates concatenation between the two strings. And if you color them, and you can see you can recover from this graph, you can recover both genomes from this graph. And this is what it looks like in a larger scale. This is a real assembly graph. If you ignore the small variance, you will get like this. And also come back to the C4 gene. And with the pangenome graph, how do we represent this case, this more complex case? This is a pangenome, this is a mini graph of this region. The red, the thick red line, this indicates the reference genome. The reference genome goes this way, goes to here, and then goes here. And then you can see there's copy. The C4A is here, and the C4B is here. This is on the reference. And sometimes you may have a deletion. If you go through this path, then you will have deletion. You will delete the C4B region, and so this one copy. And so for some other sample, you will have an insertion. This is one copy, the first copy, the second copy, the third copy. You will have 3 copies for this. And then this is a model, this 3 copies. In the human pangenome, we don't see any sample with 4 copies. And so this is 3 copies. And we can see with this pangenome graph, we can model the relationship between these genes, and we can study them. And if this is for the genes, and if we look at the other content, the repeat content of this pangenome, we will typically see this. This is this shows the count of the LOD different repeated class in the pangenome inserted into the pangenome. And this LO is mostly LO, and this is 15,000 of them. And so this is VNTR and SAGDUP. But this is by count. If you do that by the length, and you will see a different plot because the LO sequences are much shorter than the L1. Also, there's although in terms of count, there's more LO than L1, but in terms of the total length, LO is shorter. And the most striking bar is actually here, the second multiplication. Although by count, there are not a lot, but because these are all long regions, mostly the things I show in the, for example, the TCR, BCR region, and also the C4, this all go into this category, this second multiplication. This is actually contribute the most to the pangenome. And if you look at how the pangenome grow with the number of genomes, and we can see this is the bacteria, the E. coli, and how to read the curve. The x-axis is the number of genomes. You can imagine that you add one genome after another. And after you add one genome, you will see more unique content from the genome. And so this is the y-axis. This shows, in this case, the genes. And so with you're adding more and more genomes, the pangenome size will increase. The total number of genes will increase. And previously, I showed the point is around here. This 50 genes, 50 E. coli genomes. That's about 10,000. And you can see with 1,000 genes, there will be 25,000 bacteria, these genes in E. coli. It grows very fast. But in human, the curve is similar, but actually it grows much slower. This is adding about 500 genomes, and you can see it's only at about 100 megabases. The whole human genome is 3 gig. And so with 100, you only add a small fraction. And this is, if you do this, also if you see the thing that are common in half of genomes, this is only 55 megabases. And most of these are, it's actually the gene, it's actual copy number changes. These are not your unique materials. Okay, that's about the this is what's published in our paper last year. This is most about what the human pangenome looks like. Basically, at that time, we only had time to what the content of human pangenome, what's it varying, and what's not changing, what keeps stable. And so it's the HPRC, the Human Pangenome Reference Consortium, as a consortium, we are thinking what's the next step, and basically what's the future. Where we are going as a consortium. There is some important lesson we can learn from the Western genome. If you think about it, the Western genome project and the Human Reference Pangenome project, these are very similar projects in that, and both are funded by the NHGRI, and both have fully public data. And the Western genome project, I think the most important component of the Western genome project, I think that there are 2. The first is the catalog of small variants. It's basically currently everyone is using. And secondly is the tooling. And if you think what you use today, the SAM format, the VCF format, the JDK, BWA, all come from the Western genome project. Without the Western genome project, these tools actually will be, the development of these tools will be much delayed. And so what HPRC have so far is a high-quality genome. These are definitely solid data. Our tooling, we have mainstream assemblers, HiFi-SAM and workcode. These are 2 mainstream, the most used assemblers today. These are developed for the HPRC. But we can do more than that. I think the future of the HRC will soon to have 450 haplotypes by the end of this year. This is almost 4 times higher than our 5 times higher than our current 92 haplotypes. And 300 haplotypes are already available right now. Actually, you can already download them. And some more diverse populations, and also better quality. It will be better quality than the first batch. And we are also thinking about the tooling. We are thinking to develop a tool to do better SNP calling. Although this figure is already showing in the public paper, but I have to say that this is mostly a prototype. It's not ready for the end users. And this raw curve plots, this is a precision recall curve, and this is the data we can do with the pangenome. You can see the curve is above all the other two. The single sample SNP calling. You can do better single sample SNP calling. And also similarly, we can do the genotype typing, because if you call the normal structural variants with the short reads, it's actually very difficult. But if you know the structural variation is there is a structural variation, and your genotype, whether it's absence or presence in the population, and it's much easier. You can do the genotype typing. And this mostly currently people use the pangenome for that. This currently is also not that easy to use, and it also has the scaling issue, but it works. And this shows the direction we are working on. We can do the genotyping at the population scale. And also, as I said, the pangenome graph is the mainstream approach to model the pangenome, but the pangenome actually has some problems. And so the main problem is that there's not a true graph. We don't know whether the graph is correct or not. This is actually, varies us a lot, because there's no truth. We just build something beautiful, and we think that this works, but it's not really like that. And if you go deeper into the details, there's also the collapse or uncollapsed graph. I'm not going to do that. And the base level chi-square green graph, it is all the details, and also many parameters and heuristics, and also it's very slow. And I showed this C4 graph, and actually I was cheating on that. Actually, this graph is not correct. If you look at this graph, there's C4A, C4B, and on this graph, I'm labeled C4X. Because in this case, and you can see this is ABB, and so here BAB, and in this case, I can't distinguish these cases from this graph. And this is a fundamental problem with this graph. It can only model the approximate topology, but it's difficult to go into the details and what's really encoded in the graph. And so we are also thinking about the future tooling is called the ungraphed approaches. Basically, we use, say, the K-mer-based method or Barnes-Waiter transform-based method to study this region. And so in here, this is the C4 region. And so you can see from the count, you can see there's 193. There are 193 copies. Both C4A and C4B copies in total. And so here, the middle part, this is the HERB. Actually, here, this HERB is 6.4 kb insertion. There's some gene has this insertion, some don't. And also the exon 26, you can see the difference. You can distinguish between the two genes. And also, we are going about the linear reference coordinates. Currently, the current problem with BCF is there's no coordinates. For this example, this reference, and you have long insertion on the reference, and then inside the insertion, there's a SNP. And with the current method, if you do in that with the BCF, there's no way to encode this SNP, because it's not on the reference coordinates. And there's also other problem is that then the current problem, you can actually model it. You can model it, this is one allele, with G, this is one allele in the VCFL, and with A is another allele. Both of them are long insertion. But this will make it very difficult to work with, because both are long, and you cannot really see this SNP on the insertion. And currently, we are exploring this new method. This just happened several months ago. We have a tree reference. In this case, this is the thick red edge. This is a tree. You can see this is a tree. These branches here. the red edges, they form a tree. And the rest of the edges are variants. Basically, this edge, the variant edge, corresponds to this AG substitution. And this variant edge corresponds to this insertion. Then you can separate the insertions and the SNPs from that. And so this is generally the future is we have this. We want to have the better and more tooling, and also the important thing is the improved user interface. And with this tooling, we will get a map of small and structural variants. Think about the bacterial genome for small variants, and so we are doing that for all types of the variants. And this will be a great result for the implementation, and we will also try to integrate to the genome browser better. In summary, Pangenome started with the gene set analysis in bacterial genomes, and it's moved to the whole genome analysis just very recently in the past 3 years. And it made possible by high-quality assemblies, and it will provide a full category of the variants and also the tooling to study them. But there are still many practical and theoretical challenges which remain. And so with that, I will end my talk here. I want to thank my collaborators, my group members, and thank you also the audience. Thank you.
So let's open up for questions.
So we know that rare mutations are often the causing mutations for like many human diseases, especially rare diseases. But it just said that pangenome would be hard to detect those novel mutations with using short-range sequence. So my question is how useful the pangenome is for like rare disease diagnosis, especially those diseases caused by rare structure variants.
Yeah, actually, I didn't have time to elaborate this part. The pangenome is useful as a background. And if you think about how you use the binary genome in the rare disease studies, and I mean, although these more people mostly use the nomad, but you found variants and you want to see whether this is germline or I mean, say somatic or de novo mutation, and you typically check the nomads if it's present there, and so the human pangenome can work like this. And also another important reason is why we can get these better results. It's because with pangenome, we know the other sequences, we can do better read mapping. I mean, the pangenome will help to reduce the false mappings, and this will improve the variant counting accuracy. And so this will also be helpful, say, for the Mendelian disease in that world, rare disease.
Thanks.
Beautiful presentation. How often are these variants can be attributed to transposons and repeat elements? And the second question is there any evolutionary pressure on this divergence?
There are lots on these repeat elements. You can see this, I mean, the 15,000. And a lot of this is especially for the longer ones, because it is first observed. We are previously we shall really can't really do this analysis. This has been observed often, but on the biological side, but actually we haven't really studied them, and we haven't really studied the effect of them. And Team Wang probably has more insight into this category than us, than me.
And maybe also these transposons are not fully annotated yet.
It is. I think it is annotated in the. There's a VCF for these transposons. We have them. It's just we haven't really analyzed these regions.
And the evolution pressure. Do you think there's any particular pressure, such as positive selection, on this divergence?
So far, we haven't analyzed this evolution. What we have done is mostly we call them. We know they are there. So Team Wang has analyzed the methylation around these transposons, but we haven't gone further. So far, it's mostly a resource.
Okay, question on the back.
Very nice talk, Heng. So I have two questions. One is for the, you showcase the HLA region in your introduction. Can you tell us like what the pan reference helps us understand the HLA region? And the second question, specific question regarding the subtelomere region of chromosome 4, there is a 3.3 kb macrosatellite repeat, D4Z4 repeat region. In the HG38, it was a big gap. I wonder whether this region has been resolved in this new assembly approach. Thank you.
Okay, I will start with the second one. That's a good question. Previous postdoc in my group and now independent, and he's studying the subtelomeres. And firstly, it's a assembled through. I think most of the subtelomeres are difficult, but not that difficult. I think most subtelomeres are assembled through. I can't say the exact number, but the vast majority are assembled through. And there's actually lots of variability in the subtelomeres. They have its own half-lives, and actually beyond the scale of the HRA, it's much more complex than the HRA. But this is still working in progress. And as to this 3.3, that probably falls into this VNTR region. This is one of the nasty regions, the type of the nasty region in the genome. And as to the HRA, sorry, what's the question on the HRA?
How does your new assembly, what new insight helped us look into the HLA region?
Okay, I think for the HRA, I mean, it's not shown here. It's actually mostly in another paper. But we can, previously actually, this relationship, we know this is a relationship. What we don't know is actually the base pair resolution. This is the first time that we can have this like CT genes assembled through to the base pair resolution assembled through it. And so there's another group, and there's also a print on that, basically analyze this, how the habitat the difference, not just the gene regions, but it's also the repetitive region, how they differ in them.
Thank you.
Very nice talk. I have two questions. The first one is about the transposon. There is a lots of copies in the genomes. I wonder if we have new methods or new present for the transposon parts, because there are many copies, there are many variants. If we want to annotate this transposon parts, if we have any new ideas or
so far we haven't looked at the for transposon. We haven't have new classes, but for the centromeres, mostly led by the UCSC group, Karamiga, and they have new classes for the centromere repeats. They identify new subclass of centromere repeats, but actually not for the transposon. Transposon, I think we are still mostly using the repeat masker annotation, and so for human that actually still works very well.
Okay. My second question is about if we want to do a new pangenome project or a new population, what's the smallest sample size we need to do this?
The sample size. It really depends on your research question, and so far we will be getting 450 genome. I mean, this haploid genome by the end of this year, but this is actually not enough, really enough for the imputation. I mean, we can start to do imputation, but not really enough if we want to study the rare events. And I think the end of this project is targeting 1,000 haplotypes, and so I think that might not be still, I mean, big enough for the large imputation for rare studies. But if you study, say, the common ones, I mean, that may be enough. And so this really depends on what you really want to achieve these genomes. This come down to the power analysis.
Yeah, last question.
Yeah, great talk, Heng. You mentioned a lot about the future of the pangenome graph, like short-read genotyping. I just want to discuss one thing here about the closest species pangenome graph. Like, I think Eric and Peter are working on that. Do you have any idea on that? Because do you think it's possible, like, to like a human pan, like a closed species pangenome graph, within 3 years or something? What's your idea about that?
Sorry, you are thinking. Sorry, could you repeat? Sorry, could you repeat the question again?
So the closed species pangenome graph, most of the pangenome graph project is more about you do the human, the same species, different population, and you have not very little, just little divergence between the genomes. But if you give it a very large genetic difference between the, you know what I mean. Yeah.
So far, this is in the human pangenome there are 2 or 3 approaches. And so far the PGGB one that has been applied to this the larger divergence. Eric Garrison is even thinking about the apply to the whole, say the vertebrates up to fish. They want to build this one. But on the other hand, I think the practical application of that is not that clear. It's I would say it's not well studied for this. And also when there's more, the divergence higher, there will be this inversions and also complex structural variants will happen more often. And how to model that? And this come back to the question: is there's not one true graph. And so when we build a graph, what's the if we don't know what is the truth, then what's the use of the graph? I think this is an open question. So we can have some, this ongoing progress.
Great, thank you.
Thank you.
Okay, let's thank Heng again for the wonderful talk. Let's move on to the second.